Cell biology is a fascinating field that explores the intricate mechanisms and structures within cells. In this script, we will delve into various aspects of cell biology, specifically focusing on the cytoskeleton and its functions in cellular processes. The cytoskeleton, composed of three main components, namely actin filaments, microtubules, and intermediate filaments, provides structural support, aids in cell division, influences cell shape, and facilitates intracellular transport. Actin filaments, also known as microfilaments, are dynamic protein structures that contribute to cell movements, cellular protrusions, and intracellular transport. Actin polymerization and depolymerization play a vital role in cellular processes such as cell migration, muscle contraction, and cytokinesis. Actin-based cellular protrusions, such as lamellipodia and filopodia, enable cell adhesion, migration, and communication with the extracellular environment. Actin-binding proteins regulate actin dynamics and participate in various cellular processes. Microtubules are hollow cylindrical structures composed of tubulin subunits and are involved in cell division, intracellular transport, and maintenance of cell shape. They form a dynamic network that determines the organization and positioning of organelles within cells. Microtubule dynamics refer to the constant assembly and disassembly of microtubules, which are regulated by microtubule-associated proteins. Motor proteins utilize microtubules as tracks for transport, enabling the movement of vesicles, organelles, and other cargo within cells. Intermediate filaments are a diverse group of cytoskeletal proteins that provide mechanical support and contribute to the structural integrity of cells and tissues. Mutations in intermediate filament genes can lead to a variety of human diseases known as intermediate filament-associated diseases. Examples include epidermolysis bullosa simplex, which affects the skin, and Charcot-Marie-Tooth disease, which affects peripheral nerves. These diseases highlight the crucial role of intermediate filaments in maintaining tissue integrity. Centrosomes are cellular organelles that serve as microtubule organizing centers. They are essential for proper cell division and the formation of the mitotic spindle, which segregates chromosomes during cell division. Centrosomes consist of a pair of centrioles surrounded by a matrix of proteins. Abnormal centrosome function can lead to chromosome missegregation and genomic instability, contributing to diseases such as cancer. Cytoskeletal proteins actively participate in cell signaling pathways by modulating intracellular signaling cascades. Actin filaments and microtubules contribute to the localization and activation of signaling molecules, while intermediate filaments can interact with and influence signaling complexes. Cytoskeletal rearrangements, induced by extracellular cues, regulate various cellular responses, including cell proliferation, differentiation, and apoptosis. Cilia and flagella are specialized cellular appendages involved in diverse functions, including cell motility, sensory perception, and fluid movement. Both cilia and flagella consist of microtubule-based structures known as axonemes, which extend from a basal body. The coordinated movement of microtubule doublets within the axoneme generates the bending and beating motion of cilia and flagella. Defects in cilia and flagella structure or function can lead to a group of diseases collectively known as ciliopathies. Cell migration is a complex process involving cytoskeletal rearrangements, adhesion, and signaling. Actin polymerization and actomyosin contractility drive the formation of lamellipodia and filopodia, which mediate cell protrusion and adhesion to the extracellular matrix. Integrin-based adhesion complexes facilitate cell migration by providing traction and transmitting signals to the cytoskeleton. Migration is essential for embryonic development, wound healing, and immune responses. The cytoskeleton is crucial for intracellular transport and vesicle trafficking, ensuring the efficient delivery of proteins, lipids, and organelles to their respective destinations within cells. Microtubules serve as tracks for motor proteins, such as kinesins and dyneins, which transport cargo along these structures. Actin filaments also contribute to intracellular transport by guiding vesicles through actin-rich regions. Disruptions in cytoskeletal components can impair vesicle trafficking, leading to various cellular dysfunctions. The cytoskeleton plays a pivotal role in maintaining cell shape and modulating cell adhesion. Actin filaments provide mechanical support and determine cell shape through their dynamic assembly into different structures. Intermediate filaments reinforce cell structure and contribute to tissue-specific mechanical properties. Cell adhesion complexes, formed at sites of cell-cell or cell matrix adhesion, connect the cytoskeleton to the extracellular environment and regulate cellular processes such as migration and differentiation. The cytoskeleton is crucial for establishing and maintaining cell polarity, which is essential for tissue morphogenesis during development. Actin filaments and microtubules organize into polarized structures that determine the spatial orientation and asymmetry of cells. Cytoskeletal dynamics drive the formation of apical basal polarity and contribute to processes such as epithelial cell sheet folding and organogenesis. Disruptions in cytoskeletal organization can lead to developmental defects and tissue malformation. During cell division, the cytoskeleton undergoes extensive remodeling to facilitate proper chromosome segregation and cytokinesis. Microtubules form the spindle apparatus, which orchestrates the precise movement of chromosomes during mitosis. Actin filaments and myosin contribute to the contractile ring formation at the cell equator, leading to cell cleavage and subsequent separation into two daughter cells. Failure in cytoskeletal remodeling can result in errors in cell division, leading to aneuploidy and developmental abnormalities. The cytoskeleton plays a critical role in cancer metastasis, the process by which cancer cells spread from the primary tumor to distant organs. Actin dynamics and cell migration are key components of the metastatic cascade, enabling cancer cells to invade surrounding tissues, intravasate into blood or lymph vessels, extravasate at distant sites, and establish secondary tumors. Cytoskeletal rearrangements, adhesion molecules, and signaling pathways are targeted in cancer therapies aiming to prevent or inhibit metastasis. The cytoskeleton is an important network in cells that controls many cell activities. Actin filaments, microtubules, and intermediate filaments help with cell movement, transport, and division. Studying how the cytoskeleton works can help us understand normal cell functions and develop treatments for diseases.